Hello everyone, my name is Rahul. In the previous video, we had discussed about insertion of recombinant DNA. In this video, we are going to discuss about obtaining the foreign gene product. The ultimate aim of most of the recombinant DNA technologies is to obtain a desirable protein, is to produce and obtain a desirable protein. We know the central dogma of life that DNA will produce RNA, RNA will lead to production of proteins, which is known as translation. So when we insert a recombinant DNA inside a host cell, the cell will multiply, our DNA will be amplified, and the DNA will lead to production of RNA, and RNA through translation will form the required protein, which is our product of interest. Hence, there is a need for the recombinant DNA to be expressed inside the cell. The foreign gene which gets expressed, it only happens under appropriate conditions. For different cells, for different bacteria, viruses, whatever is our host cell, we will need different optimum conditions. So the expression of foreign genes, it involves the understanding of many technical details about the given host cell, but it is beyond the scope of this discussion. So right now we will discuss about the expression of protein inside the bacteria. Before that, we have to understand what is known as a heterologous host. Heterologous host. Heterologous host means in which recombinant in which recombinant DNA is present. So any cell which contains a recombinant DNA will be known as a heterologous host. This heterologous host will produce recombinant protein. Will produce recombinant protein. So any protein that is produced by a heterologous host is known as a recombinant protein. So the cells which are harboring our genes of interest, which is harboring our recombinant DNA, it can be grown on large scale, it can be grown on a small scale. So if we are performing experiments, we will only need small scale production of recombinant protein. But if we have to market this protein, such as we have to produce insulin or any other vaccine we have to produce, so we will need to produce it on a very large scale. So the large scale production requires special vessels. To produce in large quantities, we require special vessels known as bioreactors. Bioreactors. Bioreactors are special vessels which we require to produce recombinant protein in large quantities. Now, we will discuss how bioreactors function. So, inside a bioreactor, when a bacteria is growing, which is containing our recombinant DNA, which will produce the recombinant protein, there will be many phases of growth. So, in a case of bacteria, the growth phase includes an initial lag phase when the bacterial number does not increase. Then there is a log phase where the number of bacteria increases. Then there is a stationary phase where number of bacteria remains the same, and then there is a decline phase due to death of bacteria. So there is a lag phase, there is a log or an exponential phase, there is a stationary phase, and there is a decline phase due to death of bacteria. So the most amount of recombinant protein will be produced in this log phase or the exponential phase. So we have to keep our bacteria, we have to keep our host cells in this log phase to obtain the maximum protein. To perform this, we use bioreactors. So the most commonly used bioreactors are of two types. One is known as a simple stirred tank bioreactor, and the other is known as a sparged stirred tank bioreactor. So these bioreactors, they can produce these bioreactors they can produce 100 to 1000 liters of culture. 100 to 1000 liter of culture can be processed And the bioreactors, they also help in maintaining the optimum conditions of pH, temperature, etc. So in case of a simple stirred tank bioreactor, there will be a pressure valve through which we can and meter we can change the pressure as required. There will be an acid base for pH control. We can add an acid or a base depending upon the pH requirement. We can add steam to sterilize the culture media to prevent contamination. We can add sterile air which will act as a source of oxygen. Then there will be a separate opening from where we can continuously add the culture broth. The growth of bacteria will require a certain chemicals, certain nutrients which will be present in this culture broth, so we can continuously add our culture broth. Then there is a flat bladed impeller. Impeller is for the mixing of broth. It will continuously keep on churning and mixing the broth. Then there is a foam breaker. When the broth is churned inside the bioreactor, froth will form, so this will keep on breaking the froth so that bacteria can keep on growing. Now a stirred tank bioreactor is a simpler one. It has an increased surface area for oxygen transfer. So in case of aerobic bacteria, we can use this, and the bubbles, they also increase the oxygen transfer area. So in case of bacteria or cells which require very high amount of oxygen, we can use a stirred tank bioreactor. However, a simple stirred tank bioreactor is a most commonly used type of bioreactor because it has acid base control, it has steam for sterilization, we can continuously add the culture broth. It has many advantages over this simple type of sparged stirred tank bioreactor. Now, depending upon the bioreactor used, and there can be two types of culture techniques. One is known as a continuous culture and the other is known as a batch culture. In continuous culture, we keep on adding the culture medium and simultaneously we remove the product formed. So what we do is we will keep on pumping the culture media, and from one side we will keep on extracting small samples to take out the product. Why? Because in the log phase or in the exponential phase, when there is maximum growth of bacteria, the maximum product will be formed. Maximum product is formed. However, this product is toxic to bacteria. So as the concentration of the product will increase, the bacteria will start to die. So as soon as the product is increased, stationary phase will be achieved, and the product, if it is increased still further, there will be a decline in the number of bacteria. So to keep bacteria in the log phase, what we do is we keep on extracting the product and keep on adding the culture medium. So it gets the most nutrients and simultaneously this toxic product is removed. So this is known as a culture technique. In a batch culture, medium, that is the culture medium, is added only once. The reaction takes place and product is obtained only once. At the end, only once. So continuous culture we use when we require very large amount of products. Batch culture is more suitable for products where even minimal contamination is not wanted, is not desirable. See, because in a continuous culture, we keep on adding the culture broth, there is a chance of contamination. However, in case of a batch culture, because we have once closed the lid, there will be no chance of contamination. So batch culture is more suitable for products where even minimal contamination is not desirable. And continuous culture, it is used where large amount of product is required, and so we will keep on continuously culturing the bacterial cells or the host cells. So with this, we finish off with our discussion on obtaining the foreign gene product. Whatever the gene product is, a recombinant protein, it can be obtained through the help of a bioreactor. A bioreactor will have an agitator system, an oxygen delivery system, a foam control system, a temperature control system, a pH control system, and sampling ports so that small volumes of the culture can be withdrawn periodically. We are talking about a simple stirred tank bioreactor because it is the most commonly used type of bioreactor. And the stirrer also facilitates mixing and availability of oxygen throughout the bioreactor. So with this, we finish off our discussion on obtaining the foreign gene product.